What are some words that you think of when you think of Parkinson's? Tremor. Doesn't have to be a symptom. Just what do you think of?
Think of Michael J. Fox.
Michael J. Fox. Oh, I'm gonna be embarrassed. Who gets it? Old. Easy open packs. Yeah. So someone can, even if they've got tremor or muscle issues, they can get into it. I want two more. We can do two more. Involuntary. Does anyone have any family who have Parkinson's? Know anyone? Oh, okay, that's kind of surprising because of the prevalence of it. I mean, we are going to see a lot more Alzheimer's than Parkinson's, but as the population's aging, we are going to see more and more people with Parkinson's as well. So that's something that you're just going to see more and more going on. These are all awesome. These are all perfect and absolutely what's going on. So have you guys review that dopamine synapse real quick? Take a look at that, review that while I pass this out. Did anyone not get their little pack last time? Raise your hand if you don't have a little pack. Okay. 5 cents, please. Was free if you were here. Okay, and I'm going to pass these out. I want everybody to have one of these. And my goal here is that I put this on paper for a reason. Dr. Marvinova is teaching this afternoon, right? You guys are going to have all your drugs memorized by the time she teaches, right? When she teaches, she's going to be asking you, she's just going to be flying through the drug names. So what I want is when you guys are going back and forth between my slide, her slide, and trying to listen and trying to absorb all that, if you don't remember your drug names, you're not going to be able to keep up with what she's talking about. So I have created a drug list for you to have on the table next to you, so that as she's going through the lecture and she starts talking about cinnamon, you can just look over and you're like, yep, that's cinnamon. And honestly, when I came up with this brilliant idea, it occurred to me that that's something that you might think about doing with a lot of the lectures, just making a little drug list and having it next to you. If this helps you, that might be a good strategy to try to keep it so that you know what's going on during the clinical lecture. I'm not going to put them out to you though. This was just a present. Stuffing. That information. Perfect. Okay, did everyone get both? Yeah. So there's a lot of drugs here, right? So hopefully what we can do is break this down so that these drugs make sense. And something that we're gonna see is almost every single drug class here is gonna increase dopamine signaling. And so when you start looking at, oh man, what is this drug class? What is this drug class? The first thing you're gonna do when you think of Parkinson's is, will this increase dopamine? Because if I list here Haldol, what is Haldol? It's antipsychotic, and it's a dopamine what? Agonist or antagonist? I know you can. It's an antagonist, right? Because in schizophrenia, hallucinations and delusions were because you had too much dopamine at those D2 receptors in that first pathway. And Haldol was an antagonist and blocked those receptors. Here, our problem that we're going to see is too little dopamine in motor pathways. So you're going to look, and if you forget something doesn't make sense to you, think about the fact that most of these drugs are increasing dopamine. And then those mechanisms should start kind of clicking into place when you think about it critically. Okay, so what you're going to see is you've got all these guys here that are pro-dopamine synthesis. I'm just kind of introducing them as a concept right here. And so all of these things in this first column are going to help you make more dopamine. Over here, you've got straight up receptor agonists for dopamine receptors. Then you've got some things that are kind of keeping you from breaking down dopamine, helping increase release of dopamine. And then over here, you just have a couple of agents that don't touch dopamine at all, some anticholinergics and an adenosine antagonist. So really, when you think about all of these drugs, 90% of them are going to be, that's probably not a correct percentage, but the vast majority are going to be something increasing dopamine. Okay. So just reminding you guys, all neuropsych stuff has motor, psychiatric, and cognitive. We tend to think motor when we think of Parkinson's. That's the first thing we see, that tremors, slowness, but it also has psychiatric issues as it progresses, and it has cognitive issues as it progresses. So when we looked at normal dopamine signaling, back with schizophrenia, I gave you 4 pathways. I gave you the subcortical pathway, and that one was hallucinations and delusions, right? And I gave you this prefrontal cortex pathway. And when that was messed up, that created the negative symptoms, the A's. Alogia, not ataxia. Can I only remember one A? It is very early. The things that are taken away. Then I said, when we talked about antipsychotics, we talked about 2 really difficult side effect profiles. Because with antipsychotics, we were blocking D2s. But our issue was there were a couple other pathways that had D2s. And so in the normal brain, all of this is working fine. In a brain with schizophrenia, you had too much dopamine here, not enough dopamine here. If we gave an antipsychotic to normalize, then we screwed up this one and we screwed up this one. That sound familiar? Okay. So in Parkinson's, what we're seeing is instead of the dopamine misbalances being in the subcortical and prefrontal, the dopamine misbalance is in the basal ganglia. And here, what we're seeing is decrease in dopamine levels. So when you decrease dopamine levels, the basal ganglia is not able to control movement. And all of the things that we see in terms of motor issues are generally coming from that pathway. This is just, you guys know that dopamine has GI stuff and dopamine has depression stuff and addiction stuff. So you know that this is simplifying dopamine pathways here to the motor ones that we're most noticing in Parkinson's. So what we're going to see is, even though our drugs are really, the pro-dopaminergic drugs are really designed to hit those motor signals and increase dopamine signaling, there are those other types of symptoms that weren't dopamine. And the first thing that I should have told you was that, like Alzheimer's, Parkinson's is a neurodegenerative disorder. And so with Alzheimer's, we had degeneration of acetylcholine neurons in the learning and memory and cognitive regions that then spread to other stuff. And in Parkinson's, we have neurodegeneration in motor regions and other regions, and that spreads, and we get non-dopamine symptoms as well. But the things we talked about when we were thinking about keywords for Parkinson's were movement things, right? Tremor, slowness of movement, because that's what you see when you see someone with Parkinson's. And often that's why they go in, because that's their first sign that they realize that they have it. So our dopaminergic symptoms are going to be these motor symptoms. And when we talk about motor symptoms, we kind of use the term TRAP, which is tremor, rigidity, where you can't really move your muscles. So like that thing where they like push on your hand and see if it moves. If your muscles are contracted and it won't move, you have that rigidity. Akinesia or bradykinesia, we talked about those a little bit with the antipsychotics. That was one of the A's, I remember now. I can pass that question now. So that's movement. So akinesia being like too much movement and bradykinesia being not enough movement. And when you're messing, and those are opposites, but when you're messing with the motor pathways, everything's pretty complicated. You can, if you're just messing with control, you can get various things going on. And then part of the ones that you also memorize as motor symptoms are postural symptoms and gait. But those, I wanted to make sure that was kind of clear. When you remember TRAP-G, you want to separate these dopaminergic motor symptoms from the other symptoms. And when you see, my dad ended up with Parkinson's, and one of the things that you normally, when you see these motor things, the tremor here, is one of the things is it's a motion tremor. When you have essential tremor, sorry, said that wrong, it's a resting tremor. So you could just be standing there and you're not reaching for anything, but you're tremoring. Essential tremor is different. When you're just standing there, you don't tremor, but when you reach for something, you do. So those are different things because the motor regions are doing a different thing and trying to control what's going on. So when we talk about tremor, we're talking about resting tremor. Rigidity, I already told you that's kind of the difficulty moving the muscles. Akinesia, bradykinesia. Akinesia can be more movement or less movement. Postural, it was very common. What you see is a bending over, kind of a stooping over posture, and a shuffle. Gait is the walking, so like a shuffling of the feet, not pulling the feet up with the knees, but kind of just sliding them along and bent over. And so that's kind of like the key things that you can see from the outside when you're looking at someone are these motor symptoms. However, the other ones are just as important if you are a patient, because you can have these motor symptoms, but if you have these other ones that aren't treated, you're still suffering the effects of the disorder. So as it spreads and you get more systems involved in neurodegeneration. You're gonna see some symptoms of cognitive decline. Also memory loss. What did that sound like? Alzheimer's? Cognitive decline and memory loss? Because as this spreads, like every neurodegenerative disorder, it starts in a certain group of cells with certain functions, but then it kind of, as things get worse and worse, starts causing other problems. So you've got cognitive decline and memory loss, depression and anxiety, and just like with Alzheimer's, depression and anxiety are very key, but they're not necessarily a reactive response to the disorder. They often, people will have a reactive response. Like if you have been diagnosed with Alzheimer's or Parkinson's, having anxiety or depression due to that is not surprising, right? That would be a kind of a natural response. But we're talking here about the type of anxiety and depression that's coming from those neurotransmitter imbalances that you would be seeing when you're looking at general anxiety disorder or depressive disorder. Because you're messing with serotonin and you're decreasing dopamine. So having those mood disorders and anxiety makes a lot of sense. Sleep, serotonin has a big factor in sleep, and so that just is disrupted. I think one of the things to mention, a lot of times when we talk about neurological disorders, we give you these opposite side effects, right? Or opposite effects, like you could have akinesia or bradykinesia. Well, akinesia is not over movement, it's stopping of movement. So it's stopping or slowing. I apologize, I said that wrong and I'm glad that I just looked back at my slide. A always means stop or loss, and Brady means slow, right? So it's not an over movement. It's always either a slowing or stopping. But something like insomnia or sleepiness, and we're saying the disease can give you either, but they're opposites. When you talk about neuro stuff, sometimes it's really clear this pathway is increased or decreased, right? Could give you insomnia, this other pathway, or go in the opposite direction on the pathway could give you sleepiness. But it's not that simple in the brain. And so a lot of times you have a control center. And so if the control center's messed up, then it can just be not allowing things to be processed correctly. And if in your brain that incorrect processing makes you have insomnia, in someone else's brain, the incorrect processing may make them sleepier. So when you think about it, it's not like opposite things. It's not totally like opposite things are happening because it's all just coming from control being messed up. Does that kind of make sense? Okay. You can also see one of the first things that you see is loss of smell or decrease in smell, the sense of smell. And you can also have neurologically based pain, a lot of fatigue, autonomic dysfunction, and Dean Marvinova is really going to go in on the autonomic dysfunction, GI dysfunction, and then the P for postural instability I stuck over here because it isn't a dopamine symptom, but I wanted to give you that trap thing to remember. Are there any questions on this? Okay, so we're seeing from the outside when we look at someone, these motor dopaminergic symptoms, those are that is motor dopaminergic pathways, the main pathway that's degenerating. But then as we get further degeneration, we can see some of these other pathways being involved. And even arguably some pathways like smell might also be coming on early. So what we see when we look at a brain that has Parkinson's disease is we see Lewy bodies. And what Lewy bodies are is they look like they're basically just this folded clump of alpha-synuclein, which is just a protein. And it's sticky, just like when we talked about, remember when we talked about Alzheimer's, we talked about beta amyloid 42 sticking together into plaques. You remember that. I know you do. Here, we're talking about these Lewy bodies where we have this alpha-synuclein sticking together. And when it sticks together, it also starts grabbing membrane fragments, vesicles, other organelles, and it just starts grabbing intracellular stuff and creating this blob. And once you do that, your neuron's not going to live, right? You can't function like that. So normally what alpha-synuclein does is it's really just a factor regulating neurotransmitter release. And here we're talking about dopaminergic cells that are having this problem. So this is misfolding in dopaminergic neurons. And you're not expected to know all the pathways here or what this diagram means, but it's just kind of this blue part is alpha-synuclein. And when a vesicle wants to dock to release neurotransmitter, it has to use this alpha-synuclein to bring it and grab it and dock it to the membrane. And then once it's docked at the membrane and everything's released, then it comes and the alpha-synuclein is going to pop back off again and start over. So it goes fold, unfold, fold, unfold. That's important because that's telling you, yeah, this can sit in different states. It can sit in a folded state. It can sit in an unfolded state. But if you keep it folded and you keep it inappropriately folded, then it's not going to be able to release neurotransmitter anymore. And then it grabs all this other stuff, intracellular components, and you've got neurodegeneration. And this is primarily occurring in those dopaminergic neurons, particularly thinking about those motor pathways. Are we good on this one? So that alpha-synuclein, like what is it actually doing? There's a ton of things it does. And that's all you need to know. There's a whole field of research, and there are just a lot of ways that this can kill cells. And it's messing with those vesicles. It can grab onto membranes. It can cause calcium dysregulation for excitotoxicity, oxidative stress. It's misfolding proteins, so it can cause more protein degradation. Tons of things. And so that's one of the reasons that while we know what's going on in Parkinson's, we don't really know what's going on. Like we can say, oh, this alpha-synuclein is a drug target, but what is it doing? If we don't know what it's doing, then it's not a great target yet. All right. So one of the things that's super interesting about Parkinson's is it can actually spread from neuron to neuron. And we kind of talked about that as well with Alzheimer's. And it's really interesting to me, the similarities between different neurodegenerative disorders. Like with Alzheimer's, you had the beta amyloid 42 and tau, phosphorylated tau proteins. And we knew that if you took a cell with those Alzheimer's neurons and put it in a dish with healthy neurons, they would start misfolding. And this is the same case here. You can take cells that have these Lewy bodies, put them in a plate with normal, healthy dopaminergic cells, and they might start forming Lewy bodies. So the spread of it makes a lot of sense, and the way that, if it's going from neuron to neuron, the way that symptoms start progressing makes sense. Because if you're starting it in one place, those are the symptoms you're gonna get, and as it spreads, you're gonna get more and more symptoms from the brain regions that it's now affecting. Do you guys know the term prion? Most people do, but I will just say, just in case, a prion is any protein that is like a protein that you would normally have in your body, but it misfolds, and the way it misfolds will make it grab other proteins and misfold them. So now they're gonna grab other proteins and misfold them. So it's basically like infectious, but it's not an organism, it's just a misfolded protein in your body. So Dean Marvanova is going to talk about the symptom progression, but I want to tell you, like, we know it spreads in a prion-like manner. There are two main theories. One is that it's brain first, that we're seeing the symptoms begin in the olfactory bulbs and move into the motor and then spread from there. And the other theory is a GI theory, which is newer than the brain first theory. And that's saying that it's actually starting in the GI tract and then coming to your brain through your vagal nerve. And so those are different, but there's also nothing saying that it can't be one way in one person and another way in someone else, or both ways, right? There's nothing that is like that has ruled out one of these yet. They're both really promising. So I hate to give figures and not describe what they are meaning. I'm sorry I did that on the last slide. So what this is showing is the GI tract, and it's showing that you can have these misfolded proteins in the GI tract that go up through the vagal, up through the brainstem, along the areas that, so when it goes up to the brainstem, you've got autonomic, olfactory things, goes up through the sleep and motor things, and then further to those emotional and cognitive things. So this theory well explains the progression of symptoms. Well, both theories explain the progression of symptoms if you have them just start spreading from the brainstem and olfactory.
We might cover this in infectious diseases, but if a prion spreads, it's misfolding, could you have an infectious type of, like an interpersonal infectious type of Parkinson's?
So if these spread through misfolding, could you have an infectious type of Parkinson's? And I'm gonna say yes, but. There are prion disorders. Usually the only way that we know that you can get prion disorders from another person or an animal is eating tissue from them. And people in some societies that do ritual like eating of brain tissue or CNS tissue as part of like funeral rites and showing respect to the dead can then get the prion disease from eating that tissue. And that's like a known thing. And then in England, they actually had to kill like all the cows like 20 years ago because they had a prion disease that went through all the cows because they were feeding them ground up cow. Because, that's what we do. And so they were getting the prion disease from those. Theoretically, I don't know if you did have CNS tissue, either like a brain transplant or ritual eating of the CNS tissue, if you could get Parkinson's from that. It would probably just depend on what that protein's properties are. But yes, you can get prion from other people. All right. So risk factors, you guys were right on. We've got Muhammad Ali and Michael J. Fox are the two most commonly recognized people with Parkinson's because they have come out and really made a lot of steps in educating people. The risk factors in this case, being male and having advanced age are the two major ones. Also, Parkinson's is more common in people who are Hispanic or Caucasian and less prevalent in Asian or African heritage. That is a peeve of mine, but a real one that is important to talk about. When we talk about medicine and we talk about diseases and we talk about racial background, there's a huge problem with that, which is our categorizations are so made up of who we are putting together that sometimes it's practically meaningless. So one of the things, basically when we say Asian heritage, what are we talking about? You have people from North China to Vietnam to Indonesia. And is that are they the same? Genetically, culturally, this is a very big group of people. In Africa as well, the genetics and cultures from North Africa down to South Africa, there is more difference genetically between someone from North Africa, from Morocco, to someone from South Africa than there is from someone in Morocco to Sweden. So when we say African heritage in the US, what we mean is African Americans or people who were born in the US through the slave trade. And so we're talking about people who had a West African heritage. But we have a lot of people from Africa that come to the US and they don't have that heritage necessarily, you know, they are from all over the place. And so to say African heritage, when you read that in a study, I would step back. And when you say read Asian in a study, step back. Because this really, you got to think about what they're talking about here. So that was a peep, but do you think it's real and important? Okay, you can't say no to me because I'm your teacher. It is. It's real and it's important. But these are the stats that they have done. The average age of onset is between 50 and 60 years old. We know that the age of onset was in your 20s or in your 30s, but the average age of diagnosis is 50 or 60. Because just like with Alzheimer's, this is slow neurodegeneration. So you have a lot of extra neurons and they can start degenerating and you still have some backup. So you can get a decade or two of degeneration before you start to really get those symptoms. Genes are a big deal. There are early onset genes, and that was what Michael J. Fox was suffering from, early onset. Usually early onset for Parkinson's, like Alzheimer's, are some like hardcore genes of like, you just need this one or you just need these two, and you are at huge risk. The later onset or sporadic are usually multimodal, like you have to have a bunch of different genes just come in and different environmental factors come in. And so yes, there's genetic and familial risk, but it's not an all or nothing. We also know that environmental exposure is huge. And the reason we know that is we have what we call Parkinson's belts, where all of a sudden they realized that like everyone in this one, like 3 towns got Parkinson's. And we're like, what's happening here? And they were usually people who were either exposed to a lot of herbicides and pesticides, or more commonly metal smelting plants, where there were a lot of heavy metals in the air for decades and in the drinking water. So herbicides, pesticides, metals exposures, and meth abuse, you're going to end up seeing a lot more people with Parkinson's due to use of meth as younger people, because for some reason, when meth breaks down, or one of the effects rather of meth is to cause reactive oxygen species and kill dopamine neurons. So someone who has had significant meth use can have up to 300% increased risk of Parkinson's. That's just wild. And because meth has become much more common, you're going to see a lot more people with Parkinson's due to this. And then head trauma, neurodegeneration, head trauma. One thing about dopamine neurons is they are wussies. Dopamine neurons die. Like, they're ridiculously weak neurons. And part of it's the metabolism of dopamine that's what's happening. But you've got the genetics, you've got environmental exposures. Head trauma is a huge risk as well. And just one concussion can increase your risk by 35%. And then there's some comorbid stuff. And so my husband is male, Caucasian, 54, was raised on a farm in California drinking well water with herbicides and pesticides, had three concussions and 1 coma. I'm like, can you smell this herb? I'm like, can you smell the basil? But no, I mean, I'm joking, but that is an example of someone who's at high risk. And then up here in the top right is we're looking at, we can just look at this blue one, which is Parkinson's diagnosis. And you see these age groups, incidence per 100,000 person years. And you can see that starts being diagnosed around 50 to 60 years old. Incidence increases, peaks around 80, and then goes down. So does anyone know why it actually goes down? So this is not an incidence of new diagnosis. This is an incidence of people with diagnosed Parkinson's. Why would an incidence of a disease go down? Fewer people with the disease. Why would you have fewer people with the disease? Because they're dying. Yeah. So yes, they're dying because, they're 86 and people who are 86 might die. But it also, the Parkinson's disease itself creates enough motor and other issues that it can impair life function. All right. So we're going to talk about motor circuits. We're doing great here. I'm going to make a note of where we are. Okay, so motor pathways, we already looked at that review of the motor pathways that we had seen in our schizophrenia lecture. We're looking at death of dopamine neurons in the basal ganglia, which is one of the major motor regions. And you're not going to have to memorize this diagram, but I want you to have in your mind that the basal ganglia, the brain region we're talking about, is more like a pathway. It has multiple brain regions involved. So it has dopamine neurons that have cell bodies in the substantia nigra, send axons to the striatum where they have their synapses. So as you have death of those neurons from the substantia nigra, you have less dopamine release in the striatum. So if your goal is to stop, which should be, is to stop the death, you're looking at things that might affect substantia nigra. If your goal is just to boost dopamine while you've got it, then you're looking at release methods in the striatum. So here's my diagram. This is the healthy brain. In the basal ganglia, we're looking here. This is the substantia nigra, and we're looking here in the striatum. The striatum is where everything is happening. That's the synapse. So this is that dopaminergic cell sending the axon to the striatum. And now that dopaminergic cell is going to release dopamine, and it's going to cause the whole motor control circuit to function properly. This here, a neuron that leaves the striatum controls, is like the end control for motor control here. And so everything that you're doing pharmacologically is to stabilize this GABA release. So you're starting with death in the dopaminergic cell. You're ending with what you want as stabilized GABA release. So in the healthy brain, this GABA cell is modulated by a lot of things. It's got a lot of different inputs to it. These are just two of them. So this GABA cell has dopamine inputs onto D2 receptors. These are inhibitory receptors. So you don't have to memorize GI, but they're G protein coupled receptors. GI coupled means it inhibits the neuron. You've also got cholinergic inputs onto muscarinic receptors that are ones that are excitatory. So you've got these dopamine receptors inhibiting the GABA cell, and you've got muscarinic receptors exciting it. You want those to balance. To be healthy, you want those balancing out. So you've got the right amount of dopamine, the right amount of acetylcholine, everything's going well. Here's what can go wrong. Let's start in the middle. That was when everything was okay. You had the right amount of dopamine, the right amount of acetylcholine. So you had normal controlled movements. What if you had too much dopamine, I should say, too much, or not enough acetylcholine? That balance goes off. That balance goes off, and you have too much inhibition and not enough excitation. In this case, you get uncontrolled movements. So extra movements, we'll talk about what that looks like. And that's because you increase dopamine too much. What's another way you can misbalance this? You can increase dopamine. But what if you left dopamine the same? How else could you misbalance? I heard it. You said reduce acetylcholine. So if you increase dopamine and left acetylcholine the same, you'd misbalance. If you left dopamine the same but decreased acetylcholine, you'd have the same misbalance. So whichever one of those two things is happening, you're going to see the same result. If we go the opposite way and do what's happening in Parkinson's, then we're decreasing dopamine. Our acetylcholine should stay pretty much the same, but because the dopamine is decreased, now the balance is messed up the other way. So if your dopamine to acetylcholine input is wrong, and in that your dopamine signal is down, you're going to have blocked movements and tremor. So think about the high dopamine is uncontrolled movements, so kind of extra movements, and here blocked movements and tremor from less dopamine. That's what we see when we don't treat Parkinson's. That's the disease state, is those dopaminergic neurons died. Now we don't have enough dopamine input. We also, this is what's happening when you get those extrapyramidal symptoms from antipsychotics. Because when I drew that little thing out and we had what's happening with antipsychotics, and we were blocking dopamine receptors. Remember, we blocked dopamine receptors in the motor regions with the antipsychotics. That kind of mimics what's happening in Parkinson's. We used a drug that blocked dopamine signaling, or we have a disease that causes dopamine neurons to die and blocks dopamine signaling. So when we talk about Parkinson's and schizophrenia, I want you to kind of think about it's not that simple, but consider ways in which they're two sides of the same coin in the motor pathways. What happens when you're pro-dopamine? What happens when you're anti-dopamine? And what's the difference between the diseases, but what's the difference between the drugs? All right, are we going an okay speed for this? Okay, so how do you feel about this one? If you have this bottom situation here, and this is what we're looking at when we're looking at Parkinson's. In this case, in Parkinson's, we have death of these, and so we have less dopamine. There are two things we could do with this seesaw if we want our balance back. What is the first thing we could do? More dopamine. Yeah? Get more dopamine, come back to balance. What if you're not going to increase dopamine? What's your other option? Decrease acetylcholine so that comes up. And what kinds of drugs do that? Anticholinergics or antimuscarinics. So, go to the little drug chart and go down to the last row in that drug chart. Benztropine and artane. What are they? Anticholinergics. So basically, you just said increase dopamine or use an anticholinergic, and that is all but one of the drugs that you just figured out the mechanism for. So this is just kind of saying what we already said. When you have these dopamine neurons die, you have less dopamine being released. So you get that misbalance. And I'm going to have you summarize this and we're going to take a break. So let's summarize that Parkinson's disease. What kinds of neurons die? Dopamine, right, dopaminergic. Dopamine levels are going to decrease in which region? Substantia nigra. Did I say that right? I'm good. Just because I say it, I'm going to get it wrong, right? Did I get it wrong? I did. The dopamine levels are going to decrease in the striatum. The neurons are dying in the substantia nigra. So with the neurotransmitter imbalances with Parkinson's, you don't have enough actual dopamine. You just literally don't have enough dopamine. So you have too much regular relative dopamine or too much acetylcholine relatively. When we're talking about the balance, right? You have too much relative acetylcholine. You don't have, I say relative because I want you to remember you don't have more acetylcholine. Your acetylcholine stayed the same. It was just your dopamine went down, so your balance went off. So what does this do to GABAergic levels? You have a decrease of inhibition. So that means the GABA neuron fires more. And so was there actually an increase in acetylcholine or just an imbalance between these two? Imbalance. You've got it. Okay. And you guys already answered this question for me. If you want to rebalance these, strategy 1 was to increase dopamine, and strategy 2 was to decrease cholinergic signal. Yes. So let's figure out how we're going to do that. This is the dopamine synapse. We have L-DOPA is our precursor. It gets made into dopamine by an enzyme LAADC. That's L-amino acid decarboxylase. You don't have to memorize that. To have synthesis of dopamine, that dopamine then has to go into the vesicles. That's a monoamine vesicular transporter. That goes into the vesicles, they dock, gets released. After it gets released, it's going to bind to those receptors. Then it's going to either be broken down or taken back up. So dopamine is one of those that can do either one. It has both enzymes and a reuptake transporter. And then it actually has another enzyme within the cell, MAO A and B. And so it can, what's taken, reuptaken can also be broken down here. So when we had those two MAOs, MAOA and MAOB, monoamine oxidases. What were our three monoamine neurotransmitters? Serotonin, dopamine, and norepinephrine. Good. So then, which one broke down serotonin? MAOA or B? Preferentially A. How about norepinephrine? Preferentially A. What about dopamine? Preferentially B. So we're going to want to preferentially, if we have a choice, target MAOB, but we don't necessarily have a choice with our drugs. Some of them aren't going to be real specific. Some of them might be, some of them might not be. So what are our goals then? What can we do here? If you were going to design a drug, what would that drug be for synthesis? Are you going to increase synthesis? I'm hearing something. How are we going to increase that pathway?
Being agonists for dope.
So right here, you've got a precursor, you've got an enzyme, and you've got the final product. You can increase the function of the enzyme, or you could there's only one other choice here. Get more L-DOPA. Increase the precursor. All right, so there's two ways here that you could increase dopamine synthesis. So number two is getting a little. We're going to go down to number. So we've released, and now we have two things we could do. For some reason, I numbered COMT as number two. That is not sequential, so forgive me. But what would happen if you wanted to increase dopamine within the synapse? What would you want to do to COMT? You want to stop it, right? You want to inhibit COMT, catechol-O-methyltransferase. If you wanted to stop breakdown up inside the cell, what would you want to do? Inhibit MAOs. You'd like to inhibit B, but you might not have a drug that's that specific. So number four, we've done our breakdown. What's our other option to get it? What's our other option here? We had breakdown and we had reuptake. So we've got the dopamine reuptake transporter. And what do we want to do with that? Want to inhibit. And our last thing that we could do, say we're not going to mess with dopamine at all. These first actually increase dopamine concentration. Actually increase dopamine. You guys agree? What if we go the other route? We don't increase dopamine. We go directly to the receptor. What kind of drug would we use? I heard it. Agonist, right? A dopamine receptor agonist. Preferentially here, we're going to go to D2 receptors. Although we might have some drugs that aren't completely selected for that. Okay, so you have learned all of the drugs that act at dopamine. receptors that increase the dopamine function. So here is the handout that I gave you. The first one here is L-DOPA treatments. So you've got actual L-DOPA itself, and then you've got these drugs that we're going to talk about extensively, but they basically help the L-DOPA get in. Because you can't just give L-DOPA alone. So we're gonna have these drugs on top of it. Then you've got these adjuncts for dopamine. Those are those dopamine agonists, MAO-B inhibitors. And we've got one called amantadine that's really a complex pharmacology no one super understands completely. And then on the non-dopaminergic, we've got anticholinergics, which you already figured out. And then we've got this one that's kind of funky, sits here and is a whole different mechanism that we'll talk about. So right now you have most of the drug classes. Use your neurotransmitter notebook or the fancy little free packet that I gave you to tell me what side effects you might expect if you take a dopamine agent like L-DOPA or a D2 agonist that's going to increase dopamine signaling. Okay, someone comes to me and a drug rep comes to me and says, I have this fancy new dopamine receptor agonist. It has no side effects. It's perfect. I say, have you tested it to see if it is rewarding and addictive? I ask, what are its effects on movement? Does it increase involuntary movements? What does it do to the GI? Does it cause constipation? What does it do to prolactin? If you're a nursing mom, what's going to happen to your milk supply? If it decreases prolactin, it could dry up milk. When you take it, are you going to throw up? And are you going to have hallucinations? And the drug rep goes, I always pick on drug reps. I know drug reps. Some of them are terrible. Many of them are not. When you're talking about a pro-dopaminergic drug, this is what you're considering.
So are we saying that every pro-dopaminergic drug can have those characteristics?
Yeah. So good question. We're not saying that every pro-dopaminergic drug is going to have all of these characteristics. What we're saying is when we think pro-dopaminergic, these are the checklists that we're thinking about. And so maybe something does have all of them, maybe something only has a couple of them. But those are the things that we're like, could it do this? So when you have Parkinson's drugs, they can actually cause increased reward, and people can increase addictive behaviors like gambling or other drug addictions. They can, because it's boosting the reward and addiction pathways. We increase movement, which is what we wanted, right? But they can be very constipatory. We actually, for women who give birth but then can't nurse but are making milk, you can give medications that are dopamine agonists that'll dry the milk up. And we have medications we use. These can cause a lot of nausea. And it is not unknown for Parkinson's drugs that when given too high a dose to cause psychosis, hallucinations, and delusions. So if you think about what dopamine might do, you'll probably get a lot of the know a lot of the side effects of these drugs.
Another question, so since you said that, the drug itself, it increases the reward pathway, the drug itself is not addicting, it can make other substances more addicting?
So are the dopamine agents themselves addictive? There are a lot like amphetamines and cocaine and opioids and all of that stuff increase dopamine, but they hit that reward pathway really hard. Whereas the Parkinson's drugs hit it. I think a lot of it has to do with dose. Maybe some of it has to do with some like specificities and selectivities of pathways, but I don't know. I know it can be rewarding, the drug itself. I don't know if it would ever reach the point of addiction to the drug, to the Parkinson's drug. But one of the side effects is it pumps that pathway up high enough that it's easy to become addicted to other things, is more common. But that is an excellent question for Dr. Marvinova. She would know this. Okay, so when you used your neurotransmitter notebook, you figured those were all concerns. What we see, what you memorize for Parkinson's drugs is this MPGA motor. That was one we guessed, right? Increased loose motor pathways. GI issues, including constipation, nausea, and vomiting. We already guessed that. Psychiatric, including psychosis, impulse control disorders, which are, think about reward addiction, impulse control, kind of utilizing some of the same pathways. And some autonomic stuff, which weren't ones we predicted as closely, but when you think about what am I wondering about with a dopamine drug, those are explaining most of the ADEs you'll be asked to memorize. So again, memorization sucks. Understanding rocks.
Wondering if you're giving L-DOPA, if you have to be aware, basically giving someone potentially Parkinson's, the ability to move a lot easier. And now it seems like it could potentially cause mania. So somebody suddenly going, I can move, maybe I can fly, and suicidal issues.
So the question is, okay, great, you're increasing movement, you're treating, you've got movement, you've got more ability to do things, but now you've got some psychiatric stuff going on. Could that be more dangerous to a patient? Could they like be able to act like more about on delusions and depression and stuff better because they're kind of released into more activity? That's kind of the summary of your question, right? I don't know. That is a good question for Dr. Marvinova. All right. So, super important. I am going to dance so that you remember this. And if you are on Zoom, you can't see me dance because the camera's not on. So this is how hippies dance at Dave Matthews concerts, right? Have you ever been to a Dave Matthews concert? Okay, I have not. Dr. Stamper and Dr. Fortner go every single year. Every year at the Gorge. It's like their bro date, they go every year. So dance out of Dave Matthews concert. When we talk about dyskinesias, we talk about this with schizophrenia. Dys meant bad, and kinesia meant movement. So I remember I said, you can have a lot of different bad movement. They don't all mean the same thing. So when we talked about the low dopamine symptoms from antipsychotics, we talked about extrapyramidal symptoms, EPS, We said there's not enough dopamine in these motor pathways. Our dyskinesias there were tardive dyskinesia. That one that tardy, it takes a while, it's late. We talked about akathisia, where people like difficulty standing still. Talked about tremors and rigidity. So we think about Parkinson's, we said tremors and rigidity was a symptom, low dopamine. And acute dystonia, those muscle cramps, that was one of the things we saw with Parkinson's. So this low dopamine could either be a side effect of antipsychotics or mimic some of those Parkinson's symptoms. High dopamine is what you see when you have too much Parkinson's drug. So now you've increased your Parkinson's drug, maybe you increased it a little too much. Now you're extra moving, and this is the Dave Matthews Band. So you lost control of your movements, but instead of losing control and stopping them or tremoring, now they're kind of fluid and moving. So the torso and the head and neck can writhe. It might be more fidgety. There's a lot of head bobbing and swaying. So this is super important. I will test you on this. You gotta know the difference between the low dopamine dyskinesias that are side effects of antipsychotics versus this was the side, low dopamine dyskinesias, side effects of antipsychotics versus the pro-dopamine dyskinesias that are from having too much of a Parkinson drug. And if I have to make you dance, I will. But when Dr. Fortner or Dr. Stanford come in. Mm-hmm.
So like the akathisia is like the inability to sit still.
Mm-hmm.
Okay. Like compared to fidgeting, is that also kind of the same? Or like
Akathisia and fidgeting are somewhat different. Like the akathisia is more like moving around, whereas fidgeting is more just a smaller movement. But that's a good thing to ask Dr. Marvin over, like how you would distinguish those looking at someone. Because a lot of times, like I know like neurologically what's going on, but I want to make sure that you are getting like really what you're going to see as a clinician. Okay, so I'm going to show you two videos of Michael J. Fox. He does talk in this video about, he's talking about, let me see, I'm going to do the other one first. He's going to talk about about stem cell transplants. These are pretty old, but these will absolutely show you exactly what we're talking about in the motor symptoms. And yeah. And so I'm not trying to like advocate or not advocate for anything here. I want you to see in the first video here, he has taken the medication right before he goes on. And you're gonna see the high dopamine levels, because when you take a drug, you get a peak and then you get it go down. And these are so finicky that like this L-dopa is so finicky that when you first take it, you go too high and then you go too low before you can take it again. So this is him talking while he has, right after his take.
Parkinson's is a degenerative disease. Over
So
time, the symptoms get worse.
bobbing,
People with
weaving.
Parkinson's have difficulty controlling their movements as the disease takes over the brain. It can strike as early as age 30, and there is no cure. But we believe Parkinson's can be cured within our lifetime, and we're funding the research that's going to do it. With your help, we can make it stop. I only have to be smart enough to find people that are smarter than me and keep them really close. And I've done that. And that's the first rule of being a leader, is just put your ego aside and just find people that are smarter than you. I found they exist in groves.
I remember the first,
So that's that weaving. So he took his medicine and then he went on. So this is back in the day, but there was this guy, Rush Limbaugh, who was a pundit. Somebody back there knows who Rush Limbaugh is. And you're like, ugh. And he basically came on and said that Michael J. Fox was faking it for sympathy. So yeah, pundits never say anything rude like that. So here is him. This is really interesting. So now here, he takes the medication right before he takes the interview, goes in, but it hasn't kicked in yet. So now you're gonna see when it's low. And then it's super interesting because this is only 7 minutes. We're probably gonna go 5. You're gonna see as he absorbs the drug, literally how fast it goes from the Parkinson's symptoms to the drug movement symptoms. Mhm. For him, he had early onset. Oh, the onset time for the drug. I don't know. I think it's within minutes, but I hate to say that.
trying to, was his purpose to show people, so he took the drug right before the interview.
Well, he's gonna talk about that.
Joining me now is Michael J. Fox. How are you?
I'm fine, thank you.
When you did the campaign ad for Claire McCaskill, I've interviewed you on a number of occasions. But tell us what you were experiencing that day and what we were seeing as a result.
Well, on any given day, I have 1,000 different things that I can feel, and I usually feel, I go through a million cycles over the course of a day. For example, right now, this is a dearth of medication. Not by design, I just take it and it kicks in when it kicks in. Sometimes it kicks in too hard and then you get what's called dyskinesias, which is that rocking motion.
Rush Limbaugh suggested that you had failed to take your medication intentionally. So when you did that ad, you'd be more symptomatic and therefore more sympathetic.
And the irony of it is that I was too medicated and was dyskinesic. Because the thing about being symptomatic is it's not comfortable. Nobody wants to be symptomatic. It's like wanting to hit yourself with a hammer. You know, you want at all times to be as comfortable as you can be. And at this point now, if I didn't take medication, I wouldn't be able to speak. I have a masked face. I wouldn't be able to speak and I'd lock up and freeze and I'd be able to move. So there's no time that I'm not medicated.
And when you saw Rush Limbaugh pretty much imitating you on his radio show, I know that played in
I didn't see it.
on many media outlets. You haven't seen
Is
it?
he loosening
I heard
up?
about it, but you know, it's one of those things. I heard about it.
He doesn't seem as
I just,
stiff.
my first thought was, no. No, are you kidding me? And then I thought, well, you know, I knew there'd be a swift reaction from some quarters. And you know, the thing that's, and I knew there'd be that celebrity tag, which always kills me because the people who throw that celebrity tag around are themselves such huge celebrities and incredibly well-paid celebrities. And you know, really have no more God-given right to have the platform than I do or any American does. We all have a right to speak up and say what we think is right. And we all have a right to fight for the things that we believe in. And I believe that science should move forward in this country. Science is a big part of the American story. And we need to start writing a new chapter.
I called Rush Limbaugh and he told me, I believe Democrats have a long history of using victims
So
of
he's
various
like
things
picking
as
up
political
the,
spokespeople
I mean,
because they believe they are.
he wouldn't have been able to pick that glass
untouchable,
up and
They are
take a
immune
drink
from
at the
criticism.
beginning of this
He went on to
interview,
say,
right? He
Michael
wasn't
J. Fox
really,
is stumping for
he was
Democrats in the
completely
political arena
stiff.
and is therefore open to analysis
And now look at his
and
torso.
criticism, as we all are.
His leg is
Well,
going.
first thing, he used the word victim. And in another occasion, I heard he use the word pitiable. And understand, nobody in this position wants pity. We don't want pity. I could give a damn about Rush Limbaugh's pity, right? Everyone else's pity. I'm not a victim. I'm someone who's in this situation. I think I'm in this situation along with millions of other Americans, whether it's, like I said, for Parkinson's or Alzheimer's or ALS or diabetes or spinal cord injury or what have you. And we have a right, if there's answers out there, to pursue those answers with the full support of our politicians. And so I don't need anyone's permission to do that.
What about research on adult stem
That's
cells?
fantastic.
What about research on stem cells that have been culled from embryos and may be able to be produced synthetically? In other words, this is such a political hot potato, as you well know.
But the point of it is that the cells
Now that
that will
the
not
weaving
use
is
it.
gonna start coming
that are
in.
being wasted, hundreds of thousands of cells that are left over from in vitro fertilization are being thrown away, are being wasted. They're not gonna become life. They're not gonna become life. They're being thrown away. So in that sense, people say protecting the unborn, but they're gonna be destroyed anyway. So let's use those cells to protect the unborn that are gonna be born with diabetes. They're gonna be born with genetic predisposition to Alzheimer's or Parkinson's or are gonna be injured as children and have a spinal cord injury. I mean, that's the pro-life position. Because those cells are gonna be wasted. That's what
Again,
people
I'm
need
not
to understand.
going
is that-
political with this.
Is that, where was the outcry when in vitro fertilization was started 20 years ago? Because it's been going on for 20 years. Hundreds and thousands of these cells have been destroyed every year.
Let me ask you this. Is this tough for you to do?
Yeah, it's tough.
Okay, now it
But,
kicked
you know,
in.
it's hurt my wife too. She's taking one for the team. She's got the
You see
four
that?
kids at home and take them
Within,
off to school every day and all
how
that.
far are
So-
we in here?
As your symptoms become
4
more
minutes and
severe
56
and the
seconds.
medication becomes less efficacious as the
Since you
years go
started this
by.
interview and it was
How tough is
completely
this
locked
for
up,
you
right?
to sit here and talk to me like this?
Honestly,
And now the
this
is
is,
leaving.
I mean, I really feel this, that you get in your life very few chances to make a difference. And I'm really, if this is the circumstances that make that possible, like I said, I'm grateful for it. And when I'm in my life with my kids and hanging out, it's easier. It's not, and stress is really an exacerbating factor. But having said that, yeah, it's not pretty. It's not pretty when it gets bad and I'm going to throw vanity out the window. I mean, I had enough years of people thinking I was pretty and teenage girls hanging my picture on the wall. I'm over that now. So,
So could you tell that difference? Or we're having trouble distinguishing it because it's a like really crappy video quality? Oh, you can tell me. You can tell? So he didn't go to a normal section, right? He didn't just go to like the normal medication level. He went from nothing to like too low to too high, right? And so then it's that down part of the curve that you're trying to get with the treatment. Okay, so on means high dopamine. Those are on symptoms when it's too high. Or it may be, Dr. Martvinova may speak differently about it and say that on is enough or too much. And off is always not enough. All right. Do not mess up your dyskinesias, because I will ask you about them. So I'm going to have you guys take a minute and answer these questions. Okay, so a patient presents with head bobbing and swaying. Do they have too much or too little dopamine? Swaying? Too much. So are they likely to be on haloperidol? No. Are they likely to have just taken their L-DOPA? Get too much dopamine. Might have just taken their L-DOPA and have too much. Be at the end of their L-DOPA. So they're most likely to have just taken their L-DOPA because they're having those high dopamine symptoms. Off is is when your L-DOPA wears off. Peak dose dyskinesias are when it's really high because you took the L-DOPA, and tardive dyskinesia was an antipsychotic adverse effect. So this one, he's just taken the L-DOPA. Which one does he have here? Off, peak dose, or tardive? Peak dose. Okay, a patient presents with tremor and postural instability. They're most likely to be on haloperidol, have just taken L-DOPA, or be at the end of an L-DOPA dose. So tremor and postural instability is a symptom of what? Parkinson's. And so you're going to see those Parkinson's symptoms coming through when you are wearing your L-DOPA dosing at the end, right? And so what do we call that? Off. And the patient presents with jerking and chewing motions. We talked about those. We talked about antipsychotics. They're most likely to be on haloperidol. And what would we call that? Tardive dyskinesia. Correct. Okay, so we're going to start talking about these drug classes. We're halfway through the lecture. We're starting the drug classes because I truly believe if you understand how these work, you will memorize less. So the dopamine synthesis precursor is L-DOPA. And the problem is you can't just take L-DOPA. It's not absorbed. So we are going to have to do two different things. We're going to have to add a COMT inhibitor, and we're going to have to add an LAADC inhibitor. And I'm going to tell you why, and I'm going to tell you how. So L-DOPA itself increases synaptic dopamine levels. It's a precursor for dopamine synthesis. So it's going to increase dopamine release everywhere, not just the motor regions, but everywhere. And what we care about is it's boosting the function of that motor control pathway. If we could just give L-DOPA, that would be awesome. You can also call it L-DOPA, levodopa. People call it whichever, it's just completely synonymous. One of the things Dr. Marvinova is going to talk about is you can only use it for so many years and then it'll stop working. We don't 100% know why it stops working. There's some theories like mitochondrial toxicity because of reactive oxygen species. We can't pin it down and just say this is why, but it stops working, and that's sad. Like, you don't want a drug that's going to stop working when you have a degenerative disorder. So why can't we use dopamine? It doesn't cross. We can't just give dopamine itself. It doesn't cross the blood-brain barrier. So no giving dopamine. But we can use L-DOPA. It crosses the blood-brain barrier. So that's great. But it still has problems. Now, if you give L-DOPA, it crosses, but you've got to get it absorbed, GI, before you can get it to cross the blood-brain barrier. And so it's extensively metabolized in the GI. If you take L-DOPA, almost all of it gets metabolized. There's so little that gets absorbed, so little that goes into the brain. It's awful. It also has a very short duration of action. And if you're bouncing between those on and off and peak duration symptoms, that's not good. You want the longest duration of action you can so that you can try to get in a good place. And it has a lot of side effects. So we're gonna try to figure out how to help with some of these problems that we have with L-DOPA. Here is a question. When you talked about in IST1, you talked about hypotension. And there was a drug, intropin, that you used. It can be used for hypotension, but not for Parkinson's. So look that drug up and tell me what the mechanism is. Tell me what that drug is. Everybody look it up. Can someone tell me what that drug is? Dopamine. Ridiculous, right? I'm telling you, you can't use dopamine for Parkinson's, but you do use it for hypotension. What's the difference in the action here? Parkinson's, the drug has to get where? Through the, yes, into the brain through the blood-brain barrier. For hypotension, does it have to get in the brain? It does not have to get through the blood-brain barrier. So it's okay that it doesn't. And is it IV or PO? Intropin. Injection, so you don't have to worry about GI, right? So we can use intropin for hypotension because we don't have to worry about GI, we don't have to worry about the brain. But can't use it for Parkinson's. All right, so this is my favorite part of the lecture. We've got this. This is L-DOPA. So we're gonna start, here is our L-DOPA pill. We want to get L-DOPA from the pill to the synapse where it can be made into dopamine. So the first thing you gotta do is you have to get it into your gut. Problem is, there are two enzymes in your gut that are going to mess, they're just gonna screw this whole thing up. One is COMT. You don't just have it in your synapses, you also have it in your gut. And it's gonna break the L-DOPA down into breakdown products. So you're losing L-DOPA. You've also got LAADC in your gut. That's the L-amino acid decarboxylase. That's the one that's supposed to make dopamine. And when it's in your synapse, you want it. You needed it in your synapse to make the dopamine from the L-DOPA. But you don't want it in your gut, because if it's in your gut, it makes the dopamine there. It doesn't let it get absorbed. It doesn't let it go into the brain. It just makes the dopamine in your gut. What did dopamine do in our dopaminergic effects to the gut? Constipation, nausea, although I don't know if the nausea is directly from the gut or if it's from further up, but you have a lot of GI side effects. So if you just took L-DOPA, it's gonna either be broken down or turned into dopamine in your gut. 70% of that dose is lost in your GI. 70% of it. So now 28% is absorbed, well, 30% is absorbed into the blood. So 70% stays in the gut. 30% is absorbed. But you've got COMT and L-DOPA in the blood as well. So they're breaking things down. And now only 2% of that goes through the blood-brain barrier. So 2% of your L-DOPA goes into your blood through the blood-brain barrier. That's ridiculous. We just can't do that. Once it's through the blood-brain barrier, that's when the live neurons take it up. And here you have the LAADC enzyme. Here is where you may want it to go L-DOPA to dopamine. So we have a little issue here. We don't want LAADC to work in the gut, but we want it to work in the neuron.
You know that there's like an injectable, like in being thought about, like, I would assume that would be like a.
There are definitely other methods that aren't PO for giving L-DOPA. And so trying to get past that gut stuff, but there's still breakdown in the blood as well. So do you guys agree with me that you take L-DOPA alone, it either gets broken down or made into dopamine? The majority of it. That causes side effects and it causes it not to work, right? If you take it up and you can get it into the brain, then you still need to get it into the neuron, and then you use LAADC to make dopamine. So you're looking at two different things, the enzymes in the gut, enzymes in the neuron. Okay, we got that. So I say, as a pharmacologist, let's get rid of LAADC because it's making dopamine in the GI and we're losing L-DOPA and we're getting dopamine side effects. So do you guys agree that we want to get rid of this? Right here. Like that? Got rid of LAADC here. Now we're going to get more of it going into the blood. You're still going to get GI breakdown products, but more is going to go into the blood. More in the blood means more into the brain. So if we just give LAADC in the gut only, we're going to increase our L-DOPA levels, we're going to increase our dopamine levels. Increase. Increase. All right. So carbidopa is the drug we give. It inhibits it in the gut. And so it's inhibiting the conversion to dopamine in the GI. Very important, in the GI. It's decreasing the oral dose of L-DOPA needed, and it's increasing the half-life of L-DOPA because it's not being broken down as quickly. So we wanted to block the LAADC here in the gut to not make dopamine in the gut. But we still want to make dopamine in the brain, right? That's the point, is to make dopamine in the brain. So we don't want carbidopa to block LAADC in the neurons. We wanted to break it peripherally, but not in the CNS. So we asked Dr. Nelson and Dr. Stamper, our med chemists, to make sure that this drug has a particular property to where it only works peripherally, doesn't work centrally. What are they doing with this drug? They're stopping it from crossing the blood-brain barrier. Right. So carbidopa has to not cross the blood-brain barrier.
The excess symptoms.
So what is the ratio for the L-DOPA to acetylcholine or dopamine to acetylcholine to get you in a good place. And that's really patient by patient. So that's one of those things that Dr. Martin Finova will talk about dosing to try to, there's not like a number. Yeah.
I know of patients that have had like extreme allergies and they carry a backpack that gives an intravenous dose all the time. I know you've got insulin calculators. They'll inject for type 1 diabetes, and just as you measure it does, it seems like.
Okay, you guys are way ahead of me, so I'll just tell you, yes, we do have some like injectable L-DOPA delivery systems, some active delivery systems. I was gonna keep that as like a secret prize, but you're already thinking ahead. Okay, so you know why you're giving L-DOPA. Do you know why you're giving carbidopa? Okay, question. What happens if you give L-DOPA without carbidopa? Just generally, big picture, L-DOPA doesn't work. What if you gave carbidopa without L-DOPA? Nothing, right? It doesn't do anything because the whole point of carbidopa was to stop L-DOPA conversion. So you're in the pharmacy and you get a prescription for carbidopa with no L-DOPA. Ding, ding, ding. You're like, why are you taking this? All right. So carbidopa is one of those drugs that's named beautifully. Carbidopa inhibits decarboxylase for L-DOPA. So that is the L-amino acid decarboxylase. Because it's really easy to be like, carbidopa, was that for Conti or LAADC? And so you're like, does it decrease breakdown or does it decrease dopamine production? Carbidopa, L-amino acid decarboxylase for L-DOPA. So that's going to inhibit it in gut and blood. Peripheral conversion increases L-DOPA concentration in the brain. So increases in the places you need it and boosts the function of that motor control pathway. Not going to give carbidopa alone, give it with L-DOPA. There's a PO tablet, Cinemet, and there's an intestinal gel pump, Duo, giving both at the same time. I like that, Duo and DOPA, looks like both. So carbidopa does not cross blood-brain barrier. Carbidopa reduces GI side effects. And it increases the dopamine half-life. And the other drug name hint is the brand name Cinemet is going to decrease emesis. So it's going to decrease GI nausea and constipation. So carbidopa inhibits L-amino acid decarboxylase. So it's going to block conversion to dopamine peripherally, and brand name Cinemet is going to reduce those GI side effects. If this crossed the blood-brain barrier and blocked here, what would happen? If it blocked this LAADC, you would stop releasing dopamine. Yeah, you'd stop producing dopamine. It'd actually be worse if it crosses the blood-brain barrier, because it might even stop dopamine being released in other places in the brain. So if I ask you the importance of whether Cinemet or carbidopa crosses the blood-brain barrier, you know that's super important. Okay, so now we have blocked this. Take a break. Super excited. Okay, come back at 10:50. All right, so we are in a spot where we came in here and we said L-DOPA is not being absorbed because when it gets, well, L-DOPA doesn't get absorbed into the blood very well because it either gets broken down by COMT or gets turned into dopamine by LAADC. What is absorbed gets broken down in the blood and we only have like 1 to 2% getting through the blood-brain barrier. So now we were very smart. We added carbidopa, which blocked L-amino acid decarboxylase, carbidopa, and it decreased the amount of dopamine, so it decreased the side effects. So it was in MET, like emesis, decreased that. So this is better. Now we only have 40% lost in the gut. 60% gets into the blood, absorbed into the bloodstream. And of that 50% of the 60 gets lost in the blood. But now we have 10% get through the blood-brain barrier. That is a big improvement from 1%. So is that good enough? Is that good enough for you? Would you stop here? No. You'd be like 10%? No, not enough. So what else could we do? Right here, inhibit COMT. Like that? Yes, you like that. Why is this not? Okay, so now we've got a COMT inhibitor added on to the LAADC inhibitor. So we give it with L-DOPA to inhibit the peripheral breakdown to waste products. That further decreases the oral dose of L-DOPA in order to get enough into the brain and further increases the half-life because it's around longer. So now we've inhibited both COMT and LAADC. The COMT inhibitors that we have are capones. So this is nice. They're pretty easy to remember. Like Al Capone. I just remember Capone because of that. So these guys inhibit COMT in the gut and blood. So peripheral blocks that from being broken, L-DOPA from being broken down, increases L-DOPA in the brain, increases synaptic dopamine levels, boosts motor control pathway function. We have entacapone or Comtan, tolcapone or Tasmar, opicapone or Ongentys. And if we add all three good ones together, carbidopa, levodopa, and entacapone, then we have stay levo. So our levodopa stays. I was a drug company drug namer. That's my next best profession. When I retire, I want to name drugs and I will name them things that make sense. So this is stay levo, that's all three of them. Entacapone is, you can also look at that, you'll remember the mechanism because it's a COMT inhibitor. Comtan. So these drugs have a few clues in them as to what their mechanism is. So Comtan and stay levo. Tolcapone does kind of cross the blood-brain barrier, and that's okay because when we talked about the synapse, that dopamine synapse, we talked about how it had COMT and it had MAO and it needed that LAADC to make dopamine. So remember, we said blocking MAO-B or blocking COMT in the synapse would both be good. We didn't mind doing that, right? Blocking both of those or either of those, we're going to increase dopamine. So when we looked at drug targets, we said blocking COMT would be fine. That's a drug target. So while most of these don't cross the blood-brain barrier, Colcapone does. Means it has better efficacy, also means it has more adverse effects. These all increase L-DOPA half-life. So now you got the LAABC inhibitor, Cinemet, you've got your COMT inhibitors, and the capones, and you've got combination, which is the Stay-Levo. And you know what needs to get through the blood-brain barrier for sure. L-DOPA. What shouldn't get through the blood-brain barrier? Carbidopa. And entacapone in those doesn't really matter. So would adding just entacapone to L-DOPA reduce nausea and vomiting? No, why not?
Which is the LAADC that needs to be inhibited for the GI effects to not happen.
You got it. It's the LAADC that's making the GI side effects. The COMT is just breaking down and getting rid of it. So if you just blocked COMT, you're still going to have GI side effects. What would happen if you gave a Parkinson's patient, and this doesn't need this diagram, this is a question that doesn't need the diagram to answer. What if you gave a Parkinson's patient haloperidol? So they come in and they're having hallucinations, and someone's like, okay, I'm just gonna give them haloperidol for the hallucinations. So what is the I'm gonna push that further. What is the physiological problem with Parkinson's? Oh, dopamine. What does haloperidol do? Inhibits dopamine receptors. So if you inhibit dopamine receptors with your haloperidol, what's that going to do to your Parkinson's? Exacerbate. Yeah. And Dr. Marfanova is going to talk about what can you do when a Parkinson's patient has hallucinations and there's a little bit of fiddling around. It's not like you never use antipsychotics, but it's definitely not something you're just going to pop on some haloperidol for. Okay, so we talked about all those L-DOPA treatments. Now we're going to go to the dopaminergic adjuncts. Super simple mechanism, dopamine receptor agonists. We give up on making L-DOPA and we just go straight for the dopamine receptor. I want to make sure that what I said isn't interpreted wrong. When I said give up on, I'm meaning mechanistically. I'm not saying that you could never give L-DOPA and one of these. I'm saying mechanistically, we're moving beyond L-DOPA and we're saying we're gonna directly activate those receptors. Generally, the focus is D2 receptors. There's like I need both hands for this. There's a bunch of dopamine receptors, but we kind of classify them as D1 type and D2 type. So generally these are D2 type. But some other ones as well. So when I wrote D2 on those motor pathways, it's because the D2 type receptors are the most important. I may, if I ask you mechanism, ask you just dopamine receptor, or I may refer to them as D2 receptors, but I'm not going to specifically go, oh, how much of this, is this also a D1? Is this also a this or that? Just know that these are, all of these have D2 activity. So you're directly increasing the receptor activation and you're boosting function. And hopefully you're doing the same thing, the same effect as if you opened dopamine itself. So we have ropinirole or Requip, rotigotine or Nupro, pramipexole or Mirapex, and apomorphine as Apoquin or Kinmobi. The CYP1A renal, that stuff is there because Dr. Marvinova is going to talk about it, and that's on her slides. And I just wanted to have it here so that when you look back and forth between my slides and hers, kind of the same, it's popping up in both places so you remember it. So did these increase dopamine concentration? No. Several people said no, so I am very happy. No, they do not increase dopamine concentration. They're just directly acting at that receptor. Are these considered pro-dopaminergic or not? So think, what did pro-dopaminergic mean? What counts as pro-neurotransmitterergic? Does it have to increase dopamine or does it just have to mimic the effect at the synapse? The effect, right? So this receptor agonist is going to make the same effect as the dopamine itself would. So it is a pro-dopaminergic agent, but it doesn't increase dopamine. Okay, are there any questions about these drugs? These are some, like, mechanistically our easiest ones, right? Because it's just like, okay, just activate the D2 receptor. I have a trick for you to remember. Apomorphine is actually the only one that I'm aware of that is injectable. And you're poking someone to inject it. I remember it. Okay, so what was another drug target? We said we wanted L-DOPA, then we was a drug target to increase synthesis. Then we had our GI drug targets of LAADC and COMT. Then we came up here and we said, well, why don't we just have a direct agonist? And there were a couple other drug targets that we said we could do something with. We said we could do MAOB. We said we could do COMT, and we said we could do reuptake transport. Am I getting them? When we brainstormed. Okay, so here are MOAs. Our MOAs of our MAOs. B was the one that was best for dopamine. Irreversible inhibitor of this enzyme, blocks its breakdown, increases dopamine in the synapse, boosts the function. We have selegiline, rasagiline, and safinamide. And if I were your drug company, drug namer, safinamide would be safinagilline. Because I would make your life easier. But I don't have any great tricks for that. You have seen several of these drugs before when you were talking about depression. MAO inhibitors, right? Did you see selegiline or an MAO inhibitor for depression? Absolutely. So these are the ones, again, this SIP I put in parentheses because that's something that Dr. Marvinov is going to talk about. Points of interest of this, selegiline is very interesting because it actually makes a metabolite of amphetamine. And you can test positive for amphetamines on this drug. And because of that, because it's inhibiting MAOs, and when we say selective for MAOB, these MAO inhibitors are not completely selective, right? So if we have a low dose, preferentially block B, we get a higher dose, we're going to get both MAOs. So if you get MAO inhibition of A and B, then if you take a drug that's sympathomimetic, and sympathetic mimetic was sympathetic nervous system. So that was you're releasing norepinephrine. Going way back, think about your ANS. You had your sympathetic and you had the parasympathetic. The sympathetic was your stress, fight or flight. Your parasympathetic was your rest and relax. Sound familiar? So sympathomimetic would be something that's mimicking or increasing your fight or flight response. Here, an MAO inhibitor that makes amphetamines is going to increase your sympathomimetic response. So any other drug that you take that causes an increase in norepinephrine, it's not going to be broken down as well. You're going to have more of a stress response. Are we good on that? I say we good on that. That's very leading. Does anyone have any questions? All right. When low dose selegiline is used, it does not significantly increase norepinephrine and serotonin. Why is that? We're talking doses. So when we talk about low versus high dose and you're a pharmacologist, you're talking about selectivity. Right, so low dose here is more selective for MAOB, whereas a high dose is going to hit all of them. Selectivity. And here's a really good one. There's selegiline that you can take PO, and there's selegiline oral disintegrating tablets. What's the difference? What is an oral disintegrating tablet? And how does it differ from taking something PO? Absorption in the mouth, right? It's buccal absorption for an oral disintegrating tablet. Whereas you take a PO, it's GI absorption, right? The amphetamines were made by first pass. So does an oral disintegrating tablet go through first pass? No. So you can avoid the amphetamine effects by using an oral disintegrating tablet. That's so cool, isn't it? I mean, we can do so many cool things to like modify how the drugs work.
Since if they take it by mouth and they get the amphetamine metabolites, do they get like the side effects of amphetamines then, or?
My understanding is that some people do actually get more activated on selegiline, but that would be dose dependent, and I'm not sure how important that is clinically. But from my reading, it does happen. Okay, so we have, when we look at this now, gone through all the L-DOPA, the dopaminergic adjuncts, and now we're gonna talk about amantadine, which is the last one on the dopaminergic adjuncts. Amantadine is a funky drug. It's got two mechanisms of action that we're gonna think about here. Amantadine can help release dopamine. That's not even written on this slide, it's written on the next slide. But it can help release dopamine, but it also does something really important in that it is an NMDA antagonist. When we talked about Alzheimer's, did we talk about an NMDA antagonist? You said, yeah. Which one on this list did we say for Alzheimer's? Memantine or Namenda, which was NMDA. So we already know one, memantine. This one's amantadine, and we're gonna talk as we go through the pain unit about dextromethorphan, ketamine, and methadone, which also are NMDA antagonists. By the time we get there, I'm gonna have you telling me, if I say which ones are NMDA antagonists, you're gonna know. So right now you've got memantine, we're gonna talk about amantadine. What's going on is we're adding another neuron here. We had the dopamine, we had the acetylcholine, we're adding a glutamate cell. This glutamatergic cell is going on to NMDA receptors. Those NMDA receptors are activating the GABA cell. So they're acting a lot like the acetylcholine did, and that they're activating the neuron. So we've got these glutamatergic inputs. In Parkinson's, we have an increased glutamate function and an increased NMDA function or activity. So if we have increased NMDA activity in Parkinson's, then we're going to want to antagonize NMDA receptors for treatment. So NMDA antagonists are going to help regulate that response. Are there questions about this? Perfectly fine. Gonna think about it a little bit, really kind of lost. And there are no thumbs, which really means you're not listening. Okay, you can think about it a little bit. Really what's happening here is you're balancing that dopamine and acetylcholine, but now I've brought in another player that's on that balance beam. So acetylcholine and NMDA are working together against dopamine. So we call this an anti-dyskinetic because it isn't just increasing dopamine and acting like the agonists or the L-DOPA. It's used for the dyskinesias. So it's an adjunct used for dyskinesias. It can increase dopamine activity, so many mechanisms, so much don't understand. And that dopamine activity is gonna work on motor symptoms, just like any pro-dopaminergic. But then the NMDA antagonism is going to help decrease the dyskinesias. And so it's used for that treatment of dyskinesias. This is the one that I think isn't gonna make a lot of sense till Dr. Marvinova talks about how you use it. So this one would always be used along with one of the other agents. Because you'd have one of the other agents that's actually a treatment agent. And then if you're getting drug-induced dyskinesias as a side effect, maybe this one can come in as an adjunct. Here I say, I'm trying to make sure I don't miss anything on this slide, just so that guys don't get confused. It's an NMDA receptor antagonist. It has a little bit of a special property in which it's more likely to block channels that are opening too frequently. So it's kind of nice because it can kind of just pull things back without totally blocking NMDA receptors. It just kind of pulls things back to normal. So here you have the pro-dopaminergic effects, you have the NMDA antagonist effects, and by doing those together, you have the adjunct action of reducing the drug-induced dyskinesias. The Dave Matthews Band. Okay. So where do each of these drugs act? Take a second, we're not going to take too long. We've only got 2 more drug classes to do, and we can do this. So take just a moment and pop these in where they fit. There's probably one that you haven't, there's 2 that you haven't talked about yet, that we haven't talked about yet. Are you using your little chart? Your helpful chart? Okay, levodopa, number 1. Where is that acting? At synthesis. So is it here, levodopa? Okay, amantadine. What did that do? NMDA antagonist. So that was acting here to block that. Pramipexole, what does that do? D2 antagonist or D2 agonist? You got it right. Somebody on this side. Concur. Is it antagonist or agonist at D2?
Agonist.
Agonist, yes. Because you want to increase dopamine signaling, you'd never want to decrease it. Selegiline, what does that do? MAO-B, right. So selegiline, could block this. Ropinirole. D2 agonist. Carbidopa. Is it peripheral or is it in the neuron? Peripheral only, because you don't want to block, so carbidopa is blocking peripheral L-amino acid decarboxylase, which decarboxylates L-dopa. So LAADC gets blocked by carbidopa. You weren't blocking this one because if you did, you wouldn't make dopamine. So it had to be the peripheral. Benztropine, what does that do? I'm pretty sure Dr. Marvinova's talked about benztropine before. Antimuscarinic, yes. So you're blocking, you're doing antimuscarinic. Rotigotine. Dopamine agonist, yeah. Entacapone. COMT inhibitor. Mostly peripheral, but there was one that did go into the neuron, but we're really focusing on the peripheral one. Apomorphine. Dopamine agonist. And the one we're going to talk about is istradefylline, which is going to be one of the next drugs that we're going to discuss. I would play this, it's really good. Uh-huh. Does carbidopa act in the blood? Yes, it does. And when I say GI, I probably should say peripheral, as in not blood-brain barrier. The majority of its action is in the GI, so I'm kind of focused on that. But yes, there is breakdown in the blood. So good question. Okay, 2 more drug classes. All right, so we only have these 2 left. You know all of the drugs up here. So let's do the antimuscarinic drugs, which you really do already know. Muscarinic receptor antagonists counterbalances the dopamine loss. It's not increasing dopamine. Does not increase dopamine. All it does is it counterbalances that. So that in the little seesaw here, your dopamine had decreased. So now if you block acetylcholine, then you get balance again. Benztropine or cogentin and trihexylphenadiol or artane are the major ones. Does not affect dopamine levels. And you are going to need to know your antimuscarinic profile, which is handily on the first page here. And your antimuscarinic profile are things that Dr. Marvinova will expect you to know. And then the last drug I'm putting in another synapse. I wanted to start you with only the dopamine and acetylcholine. So I thought putting 4 synapses in here at once would be a little much. So we had the dopamine and acetylcholine that we started with. Then we added that glutamate NMDA receptor. We're going to add one last synapse, and that's an adenosine synapse. And adenosine is a neuropeptide. It's the one that that makes you sleepy. Adenosine makes you sleepy. And so caffeine is an adenosine antagonist and wakes you up. Do you guys know what theophylline is? Somebody knows, just just knows. You didn't learn it in class. Theophylline is the type of caffeine or similar to caffeine that's in tea. So when they say decaf tea, they're really they're reducing the theophylline in it. So basically, if you see caffeine and theophylline, pretty much the same thing. So this region also had adenosine inputs, and again, we're trying to balance. So with all of these, dopamine was the only one that had an inhibitory effect onto inhibitory effect onto the GABA cell. The acetylcholine increased GABA cell activity, the glutamate increased GABA cell activity, and adenosine increases GABA cell activity. So any of the treatments here, the treatment are either to be pro-dopaminergic or decrease muscarinic, adenosine, or NMDA. So this is an adenosine A2A antagonist, counterbalances the loss of DNA inputs, of dopamine inputs, and boosts the function of that pathway. Istradefylline, I don't have any drug name helpfulness, but it is also bypassing dopamine. It's acting straight onto that GABA cell. So all three of these, acetylcholine, glutamate, and adenosine, if you think about it, are bypassing the whole dopamine effect. All of these are just counterbalancing out dopamine. And I'm gonna let you guys look at this question. The answers are further on. I wanna do one of the take-home questions with you because it's a little more important. These are an imbalance in which dopamine is decreased relative to acetylcholine. Most PD agents treat motor symptoms by increasing dopamine levels, adding a dopamine agonist, or by giving an antimuscarinic. But Dr. Marvinova is going to talk about how non-motor symptoms are treated as well. Here's some drugs and mechanism of action that you can fill in. Here is the one that we focus on this every year. We're going to focus on these combinations, Alzheimer's and Parkinson's, and the different drugs. So schizophrenia, you had high dopamine, psychosis, low dopamine cognitive deficit. If you gave an antipsychotic, that's not helping. Okay, if you gave an antipsychotic to someone with schizophrenia, what would happen? Would it treat, what would it treat? Their psychosis? Would it treat their cognitive deficit? No, wouldn't treat cognitive deficit. What if you gave an acetylcholinesterase inhibitor to someone with schizophrenia? Wouldn't really do a lot. Schizophrenia was pretty much just a dopamine. We were focusing on dopamine. Dopaminergic agent that increases dopamine, like L-DOPA. Would you want to give L-DOPA? What would L-DOPA do with someone with schizophrenia? It would increase psychosis, but it might also help the cognitive. Probably not worth it. Anticholinergics with someone with schizophrenia. not really having much of an effect on the actual treatment, but obviously any anticholinergic drug has adverse effects. So I'm not saying adverse effects here. Alzheimer's, we have low acetylcholine, caused memory impairment and cognitive deficit. So if we gave an antipsychotic, treat, make worse, or not much? Not much. Because this is dopamine when we're really talking about acetylcholine. So an acetylcholinesterase inhibitor, what about that for Alzheimer's? Would it treat? Acetylcholinesterase inhibitor. What does acetylcholinesterase do? It breaks down acetylcholine. So if you inhibit that, what happens to acetylcholine? It increases. Do you want to do that? In Alzheimer's, you have neurodegeneration of acetylcholine neurons. So you want to increase acetylcholine. So this acetylcholinesterase inhibitors, those are the main drugs we used. What would a dopaminergic agent, pro-dopaminergic, do for Alzheimer's? Not a lot. And what would an anticholinergic do for Alzheimer's? It would exacerbate, right? Okay, so now our final one, we're gonna do it. Parkinson's. We have low dopamine to high acetylcholine ratio. So what would an antipsychotic do here? It would exacerbate, right? What would an acetylcholinesterase inhibitor do here? It would exacerbate. Do you agree? What would a pro-dopaminergic agent do? A treat. And an anticholinergic. Treat. All right. So this is something I focus a lot on because the mechanism of the drugs and how that impacts the disease state is really comes to the front when you start talking about these. And they are often, unfortunately, diseases you might see happening together. So that means Dr. Marfanova and Dr. Beggert get to fight about it. No, they wouldn't fight. Okay. If there are any, I'll hang out for a few minutes in case there are any questions you can come up.